Welcome back. This class, we are going to talk about the immune evasion of parasites. What is immune evasion of parasites? After invading the host with normal immune function, some parasites can escape the immune attack of the host and continue to live. This phenomenon is called immune escape or immune evasion. So what strategies of evasion mechanism that induced by parasites? They will include intracellular location and seclusion of parasites, and the changes in surface antigens, such as antigen shedding, antigenic variation, antigenic mimicry or masking, and suppress of host immune response. First is intracellular location and seclusion of parasites. The parasites generally have a more fixed parasite site. Some parasites live in cells, tissues, or lumens, and have unique physiological barriers that insulate them from the immune system and thus allow them to escape immune attack from their host. So the second, the changes in surface antigen. The first aspect is antigen shedding. The surface membrane of the worm is in a state of constant shedding and updating, making the antibodies that bind to the surface membrane fall off accordingly. The second aspect is antigenic variation. The parasite at different stages of development generally have stage-specific antigen. Even at the same stage of development, some species may undergo antigenic changes. For example, the glycoprotein antigen on the surface of trypanosoma can be constantly updated to avoid immune attack by the host. The third aspect about the changes in surface antigen is antigenic mimicry or masking. Some parasites express components called molecular mimicry that are similar to the host tissue. Some parasites can bind components of the host to the surface of the body, forming antigen disguise. So, the last aspect is the suppresses host immune response. Some parasites have antigens that directly induce immunosuppression in the host. They will include depletion of specific B-cell clones, induction and activation of Treg cells, and insect-derived lymphocytotoxic factor, and blocking the production of antibodies. The first aspect about the depletion of specific B-cell clones. Some parasitic infections can induce the host to produce hyperimmunoglobulin, suggesting the activation of polyclonal B cells and the production of a large number of antibodies with no obvious protective effect. At the late stage of infection, despite antigen stimulation, B cells could not secrete antibodies, indicating that the activation of polyclonal B cells led to the depletion of specific B cells that could react with antigens, which inhibit the host immune response and even caused secondary immune deficiency. The next is about the induction and activation of Treg cells. Treg cell activation can inhibit the differentiation and proliferation of immunoreactive cells. Animal experiments have confirmed that mice infected with leishmaniasis, trypanosoma, and Schistosoma japonicum produce a large number of Treg cells, leading to immunosuppression, which can reduce immunopathological damage, and may also help the parasite escape immune attack from the host. So the next is about insect-derived lymphocyte toxic factor. In the secretion of parasites, feeds, some components have direct toxic effect of blood cells known as or inhibits lymphocyte activation, such as liver, feces, trematoder secretion, which can make the lymphatic aggregations. So the Trypanosoma cruzi parasites secretion of proteins can decompose directly attached to the insect of the body surface antibody. The Fc side falls off to activate complement. The release of these lymphocyte toxic factors by the parasite is also an important mechanism for immune escape. Blocking the production of antibodies. Some antibodies that bind to the surface of the worms are not insecticidal, but can block the binding of the insecticidal antibodies to antigens. Some antibodies, called blocking antibodies, have been confirmed to exist in hosts infected with Schistosoma, mungine, and Trichinella spiralis. Okay, this class we studied the strategies of evasion mechanisms of parasites. Next time, we will introduce the development of the vaccine. Thank you. See you.